Welcome to this first of 4 videos on eukaryotic cells. As soon as I say eukaryotic cells, it may be that you're thinking, oh, those are the cells that have nuclei. Well, it's a little bit more than that. If I draw one up here like this, and I put a nucleus in it, you'd recognize that as a eukaryotic cell. In contrast to those, prokaryotic cells don't have nuclei. The term eukaryotic and prokaryotic are based on the nucleus. This part here, karyon, is actually Greek for granule. So I guess when they were looking at cells originally, they say, well, some cells have these granules in it and some cells don't. So these ones are which is a prefix, it means true, cells that have true granules, so they're referring to the nucleus. But realistically, eukaryotic cells aren't just cells that have a nucleus and prokaryotic cells lack that nucleus. The difference is really that eukaryotic cells have cell organelles that have membranes. This line that I drew here represents a nuclear membrane. And there are other membranes inside this cell as well. And prokaryotic cells, in contrast, don't have any structures that have membranes. In this video, our topic is going to be just the nucleus. We're just going to focus on the nucleus itself. In the next one, we're going to deal with a lot of the other type of membranous structures that are in eukaryotic cells. And also, actually, the third video as well. In the fourth video in this series, we'll deal with some of the cell structures that are present in both prokaryotic cells and eukaryotic cells that don't have membranes at all. So, what we've got here, let me just get rid of that. We've got the cell membrane. I'll just abbreviate things. CM can be cell membrane. Cytoplasm, nuclear membrane, and the fluid of the nucleus, nucleoplasm. The nuclear membrane is sometimes called a nuclear envelope. Because it envelops a region which is the nucleus of the cell. What we have with a nucleus is the control center. This part of the cell, inside here, contains the biochemical machinery, if you like, that has the ability to control everything that goes on in the cell. The way it does that is through chemical communication. But we have to look more closely at its structure before we can understand any of that chemical communication. So I'm going to take this nucleus and I'm going to represent it over here. And for the sake of the diagram, I'm going to suggest that I was able to slice the nucleus. It's spherical in structure, but if I were to take it and slice it through like that, and we're just looking at one level, so we look at one slice through the nucleus, we might see something that looks like this. You just give me a second here and I'll draw it. Okay, so what we have here is the nucleus itself, the nuclear envelope, or the nuclear membrane, the nucleoplasm in here, and we've got holes in it. Nuclear pores. What's inside here, this is where the chromosomes are. So we remember DNA is that double helix structure. So I'll just draw one like that. There's our DNAs in here. And the DNA is the genetic material that can control what goes on in cells. And I said that it's all done through chemical communication. Well, that chemical communication occurs this way, things can enter and leave these nuclear pores. It's important to notice a couple of things here. This nuclear envelope is actually a double layer. So we have an outer membrane and an inner nuclear membrane. Both of these membranes are phospholipid bilayers. Both of these membranes have different proteins in them. So this one here, for example, the outer membrane, has proteins which allows it to interact with the cytoplasm. So this is the cytoplasm here. And the composition of the cytoplasm, there's different chemicals in it, and it interacts with that membrane. The inner one interacts with the nucleoplasm. So the inner membrane here has a different chemical structure because of the different arrangements of proteins, and it reacts or interacts with the nucleoplasm, the chemistry, the chemicals that are inside the nucleus. So it's actually fairly complex. I'll mention another thing here. It's a little bit interesting. This inner membrane is actually lined with a certain type of protein which is not on the outside. This protein is called a lamina protein. Lamina is a word that means layer. So it just layers the inside of it. And what the lamina protein does is it sort of keeps the integrity of the nucleus. Otherwise it could collapse or something like that. But it actually keeps it in a spherical condition so that the nucleoplasm retains a certain volume and that sort of thing. It is believed that part of the aging in cells is because the lamina protein breaks down. So this integrity of the nucleus starts to diminish. Therefore, the ability of the nucleus to communicate with the rest of the cell diminishes as well. So maybe we should look at too, what type of things go in and out of a nucleus. Well, we know that DNA controls cell reactions, and it does it by making proteins. So in that process, we haven't said very much about RNA yet, but RNA is made in the nucleus and it actually functions in the cytoplasm. That's a topic of protein synthesis, which is an entirely different video series. So RNA goes out, but to make the RNA, we have to be able to have nucleotides. So nucleotides have to be able to enter the nucleus. So the nucleotides come in, they get assembled into RNA molecules, and they go out. That whole thing requires enzymes, so there has to be, enzymes have to be able to get in as well. Enzymes are made in the cytoplasm. If they're made at ribosomes in the cytoplasm. And once they're made, if they're the kind of enzymes that are used in the nucleus, they have to be able to get through the nuclear pores. So you recognize too that the nuclear pores themselves are highly specialized. And I'll just put some blue blobs on the edges of them here because the nuclear pores are lined with proteins. And that's not a big surprise either because the membranes themselves have got proteins embedded in them, and what we have at the nuclear pores is clusters of proteins which surround an opening, and because the because the proteins have got that three-dimensional shape, they've got a certain chemical configuration and a certain set of R groups, they can control what goes in and what goes out. So they're sort of like the gatekeepers for the materials that go in and out of the nucleus. That's all I want to say in this one. That sets the stage for the next video, the second one in this series. Thank you very much.